Okay, so now we're moving on to leukocytes, our other formed element that we have in our body, in our blood, whose primary role is to help defend our body. If you remember from when we talked about the formed elements to begin with, this is our only formed element that's considered to be a complete or whole cell. So it does have a nucleus and the usual organelles. But when we're talking about the number of cells in our blood volume, it actually only accounts for less than 1%. So they're far, far, far less numerous than our erythrocytes that we have. And again, their function is to help fight against diseases. If we're trying to fight against diseases or protect the body from bacteria and viruses that are not present in the blood, then we need to be able to get our leukocytes from our bloodstream to the tissue where the site of the bacteria or viruses or parasites, whatever it is, may be. So leukocytes are unique in that they are actually able to leave our bloodstream by a process called diapedesis. And then once they leave the bloodstream, they move through our tissues by amoeboid motion and positive chemotaxis. So positive chemotaxis is basically when we have damage to our tissues, the tissues will release chemicals, right? So we have a high concentration of chemicals being released right where the damaged site is. So we can have cells or bacteria use this as a sense of direction. They can move towards an increasing concentration gradient of chemicals, and that's how they know where to go once they leave the bloodstream, and that's called positive chemotaxis. Okay, so pretty much we have a steady amount, again, not a lot, only about 1% of all of our blood volume is leukocytes, but we always have leukocytes there ready to be mobilized when we need them. However, we can expect that we would increase the amount of leukocytes in a process called leukocytosis in the event that we have some sort of infection. When we're talking about leukocytes, I mentioned before that there's more than one type. We actually have 5 main types of leukocytes. We divide these 5 types up into 2 broad categories. So granulocytes or agranulocytes. So basically the 2 categories are whether or not they have visible granules within their cytoplasm. So granulocytes do, agranulocytes do not have these granules. And then if we're talking about which leukocytes belong to the granulocyte category, that would include our neutrophils, eosinophils, and basophils. Our agranulocytes are our lymphocytes and our monocytes. I would also recommend that you understand that these leukocytes, the specific types, are not present in the same quantities. So we actually have huge amounts of neutrophils and hardly any basophils at our body at any time. So order of abundance is always a good thing to know. There is a mnemonic for it, and it is never let monkeys eat bananas. So neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Neutrophils are in the highest abundance, and basophils are in the lowest abundance. So this is a good illustration of that right now. So if we take our leukocytes and we did a differential white blood cell count to see the quantity or the percentage of the specific leukocytes within our blood, we would typically see neutrophils representing anywhere from 50 to 70% of the leukocytes. Next would come our lymphocytes at 25 to 45%. Then our eosinophils coming in at only 2 to 4%. And then, it's monocytes. Then coming in is monocytes at 3 to 8%, eosinophils 2 to 4, and then very, very few basophils at 0.5 to 1%. And here's an illustration of what they look like under the microscope. So they do look very, very distinct from one another. Neutrophils are right here. We'll talk about how to distinguish between them in lab if you need to. And their uniqueness in that they typically have a lobed nuclei. Eosinophils are here, and then basophils are here, all three of which are granulocytes. So we can see the granules in the cytoplasm in the back, the little purple dots. And then we have our two agranulocytes, which are lacking or missing the granules. So that would be our lymphocyte and our monocyte. Okay, so granulocytes, again, there's three types. We have neutrophils, eosinophils, and basophils. They're all roughly spherical in shape. They're all larger most of the time than our red blood cells or erythrocytes. And I mentioned previously, but they have a characteristic lobed nuclei, and I'll show you what that means in a second, as well as those granules. Some of them or all of them are phagocytic to some degree, meaning that they can actually go in and engulf some foreign debris and help degrade it. So let's start with our first granulocyte, which is a neutrophil. Again, this is our most numerous white blood cell that we have, accounting for anywhere from 50 to 70% of our leukocyte population. They're typically about twice the size of any erythrocyte, and those granules that they have in their cytoplasm can stain with both acidic and basic dyes. Really important, though, is that the granules that are in neutrophils, they actually contain these proteins called defensins. And these are antimicrobial proteins, and they also sometimes can contain hydrolytic enzymes that can go in and help break down any of the foreign microbes that we have. The nuclei can have anywhere from 3 to 6 lobes. Let me see if I put it. I did. Also, neutrophils, they're very phagocytic, so they're referred to as bacteria slayers. So they will actually go on and pretty much engulf and kill any microbe that we have. They are able to synthesize an oxidizing substance, so something like bleach or hydrogen peroxide, and that's what allows them to go in and basically destroy the microbe itself. And the process of doing that using bleach or hydrogen peroxide is called a respiratory burst. A lot of times these granules that contain those defensin molecules can merge with a phagosome, in which case they form these spears, and these spears are going to poke holes in the membrane of the microbe. And if we poke holes in the plasma membrane of, say, a bacterial cell, basically it's going to cause it to lyse because it can no longer maintain its internal environment. So here's a picture of a neutrophil. And again, the dark purple staining is the nucleus. So when we're talking about lobes, it's multi-lobe. So this one I can see a lobe here, and a lobe here. But they can have anywhere from like 3 to 6 lobes. Our next granulocyte is eosinophils. And remember, they're only accounting for about 2 to 4% of all the leukocytes that we have. But they're still about the same size as the neutrophil itself. Their nucleus is also lobed, but it specifically has 2 lobes. And these 2 lobes are usually connected by like a little band. So it kind of looks like earmuffs when you're looking at it under the microscope. Their granules are going to contain a lot of digestive enzymes, and these digestive enzymes are really good at basically helping protect against parasitic worms, more specific. So when we looked at our neutrophils, they're less specific. They're bacteria, but pretty much any bacteria. When we're talking about eosinophils, they're really just focusing on destruction of parasitic worms, which is why we don't have huge amounts of eosinophils floating around all the time. So things like flatworms, like tapeworms, or pinworms, or roundworms, or something like that would be where our eosinophils would come in. Here's our eosinophil, and this is what I was talking about. It's always going to have 2 lobes. Here's lobe 1, here's lobe 2. Typically there's a band connecting the 2 lobes. So if you look at it, well, this one looks upside down, but it looks like you have earmuffs or some sort of headphones or something like that. Our third granulocyte is a basophil. This is the rarest one we have. If you remember, never let monkeys eat bananas. The banana stands for basophil. So we really only find them out of all of our leukocytes in a differential panel, maybe about 0.5 to 1% of them are actually basophils. And their cytoplasm contains really, really large, coarse looking, so not smooth, granules. And these granules are going to contain histamine in them. And histamine is an inflammatory chemical, and it can act as a vasodilator, meaning that it's going to cause our blood vessels to dilate. Histamine is also going to attract other white blood cells to the area where the inflammation is going on. So if you've ever taken an antihistamine, basically you're countering the effect of histamine as an inflammatory chemical. It has a really deep purple nucleus, and the nucleus is usually U or S-shaped, and I'll show you a picture in a second. Functionally similar to mast cells, but we haven't really talked about mast cells yet. But mast cells also contain histamine, but they are not considered a leukocyte. So here's our granulocyte, and you can see that we have those really coarse looking, darkly staining granules in the background, and those are going to contain histamine. Okay, moving on to our agranulocytes, we have two main types, our lymphocytes and our monocytes, but we don't have any visible cytoplasmic granules, so that's why we call them agranulocytes. They are structurally similar to each other, but they're very functionally distinct, and they're not related when we're talking about these cell types. And if we're looking at their nucleus, they're either going to be spherical or kidney-shaped. So lymphocytes, they're going to be our second most abundant leukocyte that we have, accounting for about 25% of our white blood cells. Typically, the staining of the nucleus is going to be really large and dark purple, and when you look at the nucleus, it almost is going to take up the entire space or the cell volume itself. So it's really, really, really large. You might only be able to see a small sliver of cytoplasm because the bulk is actually going to be the nucleus itself. When we talk about lymphocytes, we'll talk about this later in the next chapter. We have two types of lymphocytes. We have T lymphocytes, which we also refer to as T cells, whose basic function in the immune response is to really act against virus-infected cells and tumor cells. Our second type of lymphocyte that we have is a B lymphocyte, or a B cell, and these cells are going to give rise to plasma cells. And plasma cells are really important because these are the cells that produce antibodies. And these antibodies eventually get released into our blood. But we'll talk about this in chapter 21, which was, I think, the next chapter that we cover. So here's our lymphocyte. And again, this large purple, this is the nucleus. So that's how you know you're looking at a lymphocyte, is if you can barely see any cytoplasm. So this is the cytoplasm here. And our final leukocyte is a monocyte. Accounts for about 3 to 8% of all the leukocytes that we have. And they are the largest white blood cell under the microscope at all. They have a really pale blue cytoplasm and a really dark staining nucleus, and typically it's purple. And if we're talking about the shape of the nucleus, it's typically a U shape or a kidney shape. So we have monocytes that constantly are leaving our circulation and entering into different tissues. When we have a monocyte that enters into tissues, it can differentiate. And it can differentiate into what we call macrophages. And macrophages are really, really phagocytic cells, and they are crucial to our body's defense against viruses and other bacterial pathogens, as well as chronic infections, including tuberculosis. But again, we'll talk more about their role when we hit chapter 21. And here's a monocyte, and we can see we have this U-shaped nucleus, kind of a distinguishing characteristic of it. Okay, production of erythrocytes is going to be called leukopoiesis. So we had erythropoiesis for red blood cells or erythrocytes. We have leukopoiesis when we're talking about leukocytes and their production. And basically, their production is stimulated by chemical messengers that are being released from red bone marrow or from mature white blood cells themselves. So our chemical messengers can either be interleukins, and typically these are numbered like IL-3, IL-5, things like that. But most are going to actually be CSFs, or what we call colony stimulating factors. And they're going to be named for whatever leukocyte population they actually stimulate. So for example, granulocyte CSF or GCSF, something like that. All leukocytes are going to originate from that same hemocytoblast stem cell that our erythrocyte originally originated from, and it's going to branch into two pathways. So the hematopoietic stem cell is going to branch one way into a lymphoid stem cell line, and the other branch of it is going to be a myeloid stem cell line. The lymphoid stem cell line is the one that's going to be the pathway to produce a lymphocyte. The myeloid stem cell line produces all other formed elements. So if you remember when we talked about erythropoiesis, all of our erythrocytes were originally progenerating from a myeloid stem cell. Same thing when we're talking about all of our leukocytes, the same myeloid stem cell line with the exception of lymphocytes. So lymphocytes originate from the lymphoid stem cell line. So we have the same process. It's slightly different depending on whether or not we're producing a granulocyte or an agranulocyte. So granulocyte production, again, we begin with a myoblast. Then that's going to develop into a promyelocyte, at which point it's going to accumulate lysosomes. So that's a big characteristic there. Because remember, lysosomes contain digestive enzymes in them, which we would want our white blood cells to have. Then we have myelocytes. This is where we're going to begin to accumulate granules if we're talking about a granulocyte. And then the band cells. So band cells are going to be where we are forming those lobes of our nuclei. The bands are where the lobes are actually connected. And then eventually we reach our mature granulocyte, and the nuclei are going to be segmented into those lobes before they're actually released into the bloodstream. When we're talking about storage, we actually have 10 times more granulocytes that are actually stored in our bone marrow than our blood itself, and about 3 times more white blood cells are formed than red blood cells, because remember, white blood cells have a much shorter lifespan because a lot of them die fighting, basically, as they're trying to protect our body against these foreign invaders. Agranulocyte production is a little bit different. Again, it's going to depend on whether or not our goal is to become a monocyte or a pathway is to become a lymphocyte. So monocytes are also derived from the myeloid stem cell line, just like the other 3 leukocytes we've talked about, as well as our erythrocyte. And basically, from the myeloid line, they go on to become a monoblast, a promonocyte, and a monocyte. And they do share a common precursor with neutrophils, which I'll point out to you on the flowchart on the next slide. And these can actually live for several months. So they don't have that short, couple day lifespan like our other white blood cells. Our lymphocytes, as mentioned previously, are derived from a different stem cell line, and that's going to be our lymphoid line. And depending on which type of lymphocyte we're making is going to be dependent on which precursor we start with. So we can either have our T lymphocyte precursors, which are going to give rise to our immature T lymphocytes, which are eventually going to mature into the thymus. Our B lymphocyte precursors are going to give rise to immature B lymphocytes that are going to mature within the bone marrow. So if we're talking about lymphocytes, their lifespan is different. It can vary, so anywhere from a couple hours to several decades. And here we go. Here's our hematopoietic stem cell. And again, this is the same stem cell that our erythrocyte developed from. But in this case, we're only showing the leukocyte development. So we start over here, we go to the myeloid stem cell line, and we can see we have 4 of our leukocytes that use this stem cell to undergo its developmental pathway, and that would include our eosinophils, basophils, and neutrophils, as well as our monocytes. We talked about how monocytes frequently leave the bloodstream and enter our tissues, and when they do that, they become macrophages. Then our second stem cell line is going to be our lymphoid stem cell line, at which point we produce B lymphocytes or T lymphocytes, and our B lymphocytes eventually become plasma cells as they mature in our bone marrow, and our T lymphocytes become effector T cells. So what do you need to know? I don't expect you to know the names of anything from here to here. I'm more worried about that you understand what stem cell line these leukocytes are originating from, and understand that monocytes also differentiate to become macrophages when they enter tissues. Okay, leukocyte disorders. So we can also have overproduction of white blood cells. If we have overproduction of white blood cells, they are typically abnormal. So if we're talking about leukemia, or if you've ever heard of mono, infectious mononucleosis is what mono is basically abbreviated to. And we can also have abnormally low white blood cell count, in which case we'd say you have leukopenia. And if you have leukopenia, it's normally induced. Something can be induced by a drug, anti-cancer drugs, or something like a glucocorticoid. So leukemias, this is a cancerous condition, and it's going to involve the overproduction of abnormal leukocytes. And usually it's going to involve clones of a single abnormal cell. So basically you have one abnormal cell that replicates out of control and proliferates out of control, and it's going to end up impairing the normal red bone marrow function, and it can progress pretty quickly. So they name it according to the abnormal white blood cell clone that was involved. So the myeloid leukemia is going to involve myeloblast descendants, and our lymphocytic leukemia involves our lymphocytes. So our other 4 leukocytes would be, if we have some sort of abnormal production of them, would be occurring from our myeloid leukemia. So our neutrophils, our basophils, eosinophils, and our monocytes would all be referred to as myeloid leukemia. If we're talking about our lymphocytes, it's specifically lymphocytic leukemia. When we're talking about leukemias, they're usually quickly advancing if they're going to be derived from a stem cell. And if we have acute leukemia, that's primarily going to affect children. We also have chronic leukemia, which is very slow advancing leukemia, which is going to involve proliferation of later cell stages, and this is going to be more prevalent in elderly people. But without any type of therapy, pretty much all leukemias are fatal. And again, if we're talking about leukemia, we're talking about immature or non-functional white blood cells flooding our bloodstream. We have cancerous cells that also fill our red bone marrow, which means they're going to inhibit the production of other cell lines, which can lead to anemia and bleeding. Death is usually from internal hemorrhage or overwhelming infections because we are producing non-functional white blood cells. So if we are having some sort of infection, we don't have the white blood cells there to help combat it. So again, treatments, things like radiation, drugs, and stem cell transplants. Okay, infectious mononucleosis. This is also sometimes called the kissing disease because it's very, very contagious. It's frequently seen in young adults. It's actually caused by a virus, and that virus is called Epstein-Barr virus. Pretty much how you know you have infectious mononucleosis is that you have excessive numbers of lymphocytes. And many of these lymphocytes are actually really, really, really large. And atypical. So they originally thought that the lymphocytes were monocytes because of how large they were. So that's why it's called infectious mononucleosis. But later they found out that it was actually lymphocytes, not monocytes, that the huge number of them. Symptoms, typically tired and achy. Sometimes you have a sore throat and a low-grade fever. There is absolutely no cure for infectious mononucleosis. You just have to rest and let it run its course, and typically you're fine 4 to 6 weeks later.